Good morning, everyone. We want to start now because we're already a little late. So please, everyone, quietly get the morning started. Those who move in, please quietly. Everyone else who's sitting at the edges, maybe move in a little bit because there might be more people coming in. Okay, so before we start the GAC, I'd like to welcome to the GAC number 2, comparing artificial and biological networks. Are we limited by tools, hypotheses, and data? I'm one of the organizers this year, and as there are many people today or at this conference for the first time at CCN, I'd like to generally introduce very briefly the idea of the GAC. So the GAC is motivated by the idea or by the discrepancy between a scientific ideal where everyone kind of engages all the theory and hypotheses all the time, and a practice where often labs and researchers stick with the same kind of theory and tasks and don't engage the other theories. And here GACs comes in, where here researchers with alternative theories collaborate to resolve the differences, and they come together to work out these differences and find ways to disambiguate theories. And that is in a healthy, collaborative environment. And so for this community, this has many benefits. So one of which is that today we all can learn a lot. So I'm certainly going to learn a lot now. This is a generally community-driven engagement where we want to work out the differences together. The idea is here that we can, at this convention or in this group, decide together what are the important topics we want to discuss about. And this is one of the key features of GAC. This is open and transparent, so the proposals are open, you can comment on them. This is an open kickoff workshop, and there will be a position paper. This should all be very much community-driven. So the GAC idea was introduced by Megan Peters here to CCN in 2020. We have 13 GACs so far. This is the first fully in-person GAC, with one talk as an exception, as an emergency, zooming in. But otherwise, use that opportunity. So if you disagree, agree, hit them up later at the conference and continue the discussion later. You find those former GACs recorded and live streamed, this one as well. You find all the proposals and discussions on the GAC homepage, where you can also comment on the proposals. There has been one brave commenter so far. You can continue this after this GAC. And I encourage you to do so, and the authors and organizers are encouraged to respond to that and engage with the comments. There have been, each GAC ends with a position paper in neurons, behavior, data analysis, and theory, and there have been 5 preprints so far. Lastly, I can just say GACs are great. I've been involved in one from 2 years ago. There's slow and steady progress. We're meeting on a continuous basis. Yesterday we had dinner, many of us together, deciding on how we're going to finalize the paper. So I can only say it's great, you should do it, and you should consider submitting one next year. So without further ado, you should get started and bring the interesting topics here.
Hello, everyone, and a very warm welcome to this GAC workshop. The title of our GAC is Comparing Artificial and Biological Networks. Are we limited by tools, hypotheses, or data? And we have put up a core team of several members who, as you'll see shortly, bring a very unique perspective to this question. And we have many exciting talks planned during the workshop, so in the interest of time, we'll keep this introduction very brief. So as you all might be aware, the last decade has seen a meteoric rise in research comparing artificial and biological networks. ANNs have helped usher in a new revolution of sorts in cognitive science and neuroscience. And one of the strongest success stories was the pioneering work in the domain of vision, where it was shown that deep convolution neural networks trained on behaviorally relevant goals, like object categorization, often learn internal representations that are very similar to the primate ventral visual stream, or IT. And this has inspired a lot of optimism that by comparing ANNs and brains, we can explain specific characteristics of the brain as optimized solutions for specific computational challenges faced by organisms. And the success of CNN models of vision has also been accompanied by similar success stories in other domains of cognition and perception, including audition, olfaction, language, navigation, motor behavior, and higher order cognition. Where similarly, there's been a wealth of research comparing ANNs trained to produce human-like capacities against the corresponding neural systems in the brain that support those capacities. And so as part of our GAC, we really want to address the major obstacles that might be hindering progress in this comparative analysis of artificial and biological networks. And so it would really help to articulate what are we really trying to achieve when we compare these diverse systems. And so from a neuroscientific perspective, we employ these analyses to really understand neural computations at an algorithmic level. We want to quantitatively explain the neural mechanisms that underlie these vast perceptual and cognitive abilities. And the hope is that we can meaningfully distinguish between competing hypotheses, understand the similarities and differences between different artificial neural networks and biological networks, so that we can make strong inferences about how the brain works or why the brain works the way it does. And so I want to quickly revisit the whole framework of comparative analysis, since it's the central topic of our GAC. So under this framework, we have an artificial neural network which encapsulates some hypotheses about how the brain works. Often we have a group of artificial neural networks which encapsulate competing hypotheses about how the brain works. And unlike brains, we know exactly what the network has been optimized for, what data it has been trained on, what architectural inductive biases it has, and the learning algorithm that is used for updating its weights. And so the hypotheses can be thought of as being embedded in either of these ANN elements. We also have neural data, which are empirical observations from biological networks. And then we have a set of tools and metrics that we can employ for comparing these two diverse systems. And so while this framework has been tremendously successful, partly also due to the progress in machine learning and the strong capabilities of modern artificial neural networks, here in this GAC particularly, we ask whether progress in this comparative analysis enterprise might have slowed down, and whether we might be limited by hypotheses, tools, or data in this enterprise. And so now I'll just quickly hand over the mic to Ratan, who's going to show some evidence that progress might have slowed down, and then he's going to just talk about some of the considerations that are entailed within each of these three different bits.
Okay, thank you. So, what are the ingredients of this research program, right? So, what we've done now for the purpose of this GAC is we've divided the entire thing into three groups. Data, which is the kind of neural data that you want to predict or explain. The metrics, which is what you compare, like the way you would compare biological systems with your artificial neural networks. And hypotheses, which are, in general, what we think of as base models that are used to predict the brain or behavioral data. So after some early success, we think that progress has been relatively slowing down. And why is it that we think that progress is slowing down? So there are many papers that have compiled these evidence. So here's a plot from Martin Shrimp's paper in 2021, which shows that for a while we had, as engineering benchmarks improved, which is what's shown on the y-axis, match to the brain was improving. And then at some point, it stopped, right? There are other papers that show that if anything, there might be an opposite effect. We're running into more and more artificial neural network models that come from the machine learning literature, which are doing really well at explaining engineering benchmarks like top 1% on ImageNet, but they are no longer brain-like. So why is progress slowing down? So let's come back to this Venn diagram. The goal of this GAC is not to distinguish between whether it's because of data, it's because of metrics, or hypotheses. It's probably all of the three, right? So the way we've structured this GAC is the follows. We're going to discuss each of these factors in isolation. We have a bunch of speakers who are going to present talks on each of these broad headings. For the talk sessions, you should think of them as one single lecture, with each participant discussing a slightly different version of the overall umbrella topic of like hypothesis, data, or metrics. And we will discuss all of these factors in isolation. The goal is to try and articulate what we think are the specific roadblocks, and also to try and see what could be possible ways forward. So what we have today are going to be, as I mentioned, talks and some panel Q&A per topic. We've asked all of our speakers to restrict their talks to 8 minutes, and we're going to keep them at that. And we're going to have a panel Q&A at the end. So I'm also seeing that this is a packed audience, which is great. We will ask all of you to participate. So the way you participate is the following. So this is probably the most important slide of my talk. Go to slido.com, and you have to use the code GAC2023. We are also going to be running a bunch of polls. We want to just take the temperature of the room and see where you are at. So if you keep refreshing, you'll see that the polls keep changing. So there's going to be some element of surprise. It's not that surprising. But all questions will be taken via the Slido. So everyone, please make sure that you have Slido. You should all be able to access it. If you're not, please send us an email. We will be taking those questions, and you also have the opportunity to upvote your questions. I have one request: keep your questions short, so that we can take a lot of those questions, because that really is the goal of the GAC. That's it. With that, we will start the first session, and I'll request the speakers for the first session to come up on the stage. Thank you.
So first up, we'll really kick off the hypotheses section of the workshop. Our first speaker is Aran Nayebi, who's a postdoc at MIT, and he's going to be arguing that we need new models, especially when it comes to innovations in terms of model architectures.
Great, thank you, Meenakshi and Ratan, for organizing this GAC. I'm really excited to be here today. So today I'm going to suggest that we may need a couple new ideas. And in particular, as other folks have mentioned, a lot of this is motivated in the brain score observation that as we plot ImageNet performance versus neural predictivity, different CNNs that have a reasonably high ImageNet accuracy end up predicting macaque V4 and IT quite similarly, despite having very different inductive biases. And perhaps more strikingly, recent work led by Colin Conwell in Talia Konkel's group has shown that when matching human occipitotemporal cortical responses, both CNNs and vision transformers that are functionally reasonable appear to be matching these the same, despite having vastly different inductive biases to begin with. So a natural question is, why might this lack of distinction be? And the nice thing about our field is that we can kind of enumerate this in a finite list, and they kind of come under these three headings of metrics, brain comparison data, and models. And within those, we can consider, for example, more strict mapping transform classes, or causal manipulations, or topographic metrics, and folks will get into that. And the answer is really all of these. But I guess what I'm going to show you today is an example where I keep the metrics the same. So I'm going to stick to regression, correlation-based metrics. And I'm going to keep the organism the same as well, which are going to be primates, both macaques and humans. But what I'm going to do is I'm going to make two modifications to the brain comparison data. In particular, I'm going to have the animal perform a cognitive task. This is as a difference to prior work where typically animals are passively viewing images. And moreover, I'm going to switch the brain area to be from recordings in frontal cortical areas. And what I'll show you is that when these two modifications are made, we're quickly led into a position where vastly new model ideas are needed across architectures, loss functions, and data sets. So the overarching motivation of this work is that you and I do a lot more than simply view the world. We have to predict and anticipate the actions of other objects so that we can plan our own actions. And so for us, the crux question here is what might be the neural mechanisms that enable the brain's simulation-like computations of the future state of the environment across a diverse range of settings. So to better get at this question, Rishi Rajalingam and Murdad Jazairi's group trained two macaques to play a game of Pong, where they operate a joystick to move the handle up and down, like that on the right. But the catch is that basically as the ball approaches the handle, it gets blocked by an occluder. And they had shown that there was evidence of this type of tracking of the ball state while it's occluded, its position and velocity in the underlying neural activity of dorsomedial frontal cortex. And there's about 2,000 neurons total from these areas. And we're interested not just in Pong, but more generally in naturalistic environments. How might this type of mental simulation be going on? And to get at this, we took a bunch of built and also evaluated a bunch of state-of-the-art models in their own domains. And it's about 40 models, but it comes down to about 3 different hypotheses classes. So the basic idea is you take in a scene and you predict the future either at the level of pixels, which is on the bottom here, or at the level of fixed object slots, and then you run dynamics forward with a graph neural network. But the main one I want to really particularly place focus on is the top one, which is this latent future prediction paradigm. And what that is rather than posit a particular latent state, what we're going to do is first break it down into a vision stage where we learn a partial implicit representation of the physical world by performing a challenging vision task. And the main emphasis is that this latent state is reusable. And what I mean by that is when we train this visual encoder, it can transfer to a bunch of different downstream environments and embodied control tasks. And for example, that can be done on ImageNet, but a recent data set where people wear head-mounted cameras around the world, they go about doing everyday actions. And this one is actually going to be particularly crucial as a pre-training data set that I'll show you in a minute. Now, once we have that reusable latent representation, what we're going to do then is learn dynamics that predict the future state of the environment in that latent state. And those dynamics are going to be the ones doing the explicit physical simulation. Now, the nice thing is that despite these models being very different, they all output a set of stimulated states. And because these models are pixel computable, we can evaluate them on held-out scenarios in this naturalistic 3D environment, compare them to human behaviors of yes-no object contact predictions, or also to the completely novel, to the model's Pong environment, and try to predict neural activity as well. So for now, I'm just going to focus on the neural predictivity results, and then I'll get back to human behavior at the end. Right off the bat, if we look at neural predictivity of the models, the animal-to-animal consistency is quite high. So 2 macaque DMFCs can explain each other quite well, so it's a reliable metric. The best models that are about 50% neural predictivity are these models that are pre-trained on Ego4D, these video foundation models. And in particular, they need to have dynamics, because if we just look at their fixed encoder on the left of each group, that does quite poorly. So you do need to do that future prediction in the latent space. But moreover, not all video foundation models are equal. So if we pre-train the encoder on the same data set, but use a more reinforcement learning loss as opposed to a self-supervised one, neural predictivity goes down. We're also better by doing this latent future prediction than, say, pixel-wise or object slot-based models. And finally, the main point of comparison I want to point out is that not all foundation models themselves are equal. So in particular, if we take the image foundation models, like CNNs and transformers, either trained in a self-supervised way or supervised on both ImageNet and even larger than ImageNet web-scale datasets, These are going to be representations that are going to be most useful for things like image segmentation and classification, yet the models that best match the data are actually learning representations that are most useful for downstream robotic manipulation and embodied control tasks. So just to put the numbers in context a little bit, where we're at is sort of like where we were at about 7 to 8 years ago with inferior temporal cortex, in that the best then novel CNNs were getting about 50% explained variance, and the prior more handcrafted models were getting about 25 to 30% explained variance. Now, like I said, we don't just want a model of Pong. We also want to be able to generalize to held-out scenarios in naturalistic environments. So for that, we compare to human error patterns. So basically, what they have to do is predict whether two objects will come into contact or not by viewing just a segment of a video before the actual objects collide or not. And on the x-axis here, I'm plotting that correlation to the average human response, and the y-axis is the same neural predictivity. And in particular, again, the dynamically equipped video foundation models can better match both. So the takeaway so far is that mental simulation appears to be primarily relevant to predicting the future state of the environment in a suitable latent space. This latent itself is highly constrained. It doesn't seem to consist of bespoke object slots or pay attention to fine-grained details at the level of pixels, but really has to be reusable across dynamic scenes. And so far, what we're seeing is a correspondence between being able to predict this neural and behavioral data and developing useful representations for embodied AI rather than more classic computer vision tasks like classification, segmentation on ImageNet, et cetera. But again, we're not perfect, so how do we get to better models? Well, here's a couple ideas based on the results we're seeing. One is to learn a more factorized representation that's also reusable. So in particular, we see that Oracle models that have access to more state variables do better on this data, ground truth ball variables. But moreover, what we want them is to be reusable, unlike the object slot models. So maybe ideas from cognitive science could help enable building object-centric video foundation models. The next point is maybe there could also be hierarchy and modularization of timescales in the dynamics, multiple timescales of hierarchy. And there's some evidence for this in recent work from Murdad's group in frontal cortex during reasoning. But maybe that might help leverage more diverse material properties, since currently all the models really struggle at soft body with rigid body interactions, like the drape scenario on the right here. But they do fine on rigid body support interactions. With that, I'd like to thank my collaborators, Rishi, Murdad, and Robert, and my funding sources, and we also have the preprint online for more details. Thank you.
And just a gentle reminder to the audience that we'll be deferring all questions to the end of each section, but you can all still post your questions on the Slido platform. The code is GAC 2023. And our next speaker is Tal Golan, who's an assistant professor at Ben Gurion University. And he's going to be arguing, he's going to be talking about some of the limitations of current models and how we might be able to expose them.
Thank you very much. So in this GAC, we face the question, are we limited by tools, hypotheses, or data? My answer is that we are usually limited by data. But this is a limitation we can readily overcome. And once we remove this limitation, we see that our actual limitations lie somewhere else. So why are we usually limited by data? When we test and compare neural network as models of biological vision, we typically present a set of natural images to both our candidate models and to human or animal subjects. Using natural images as test stimuli makes perfect sense. Any model of perceptual processing must consider the stimuli the brain evolved to perceive. But is it sufficient to test models exclusively on natural images, natural stimuli? The typical result we obtain when we measure the correspondence between how neural networks and brains represent natural stimuli is that of multiple distinct models that predict the neural or behavioral responses equally well. Let's take a second look at this study by Colin Conwell and colleagues. They used the NSD dataset along with an extensive set of models. Here, this is just part of their data. Comparing different architectures, they found a limited effect of architecture on model brain alignment. When they allowed for flexibility in the comparison between brains and models by means of fitting feature weights, this improved model performance across the board. But it also made model comparison even more challenging. When manipulating the training task instead of architecture, the problem persisted. We can see model brain consistency in neural networks trained, similar model brain consistency in neural networks trained on purely visual contrastive learning and neural networks trained on linking images and language. This problem is not unique to this study, it's not unique to human fMRI. We see this pattern of multiple models similarly aligned with brain data across the board, from behavioral studies to monkey electrophysiology. The way I see it, the common issue is that when we test our models solely on natural images, we test these models within their training distribution. This does not sufficiently probe the inductive biases of these models. These are the assumptions that different models make that allow them to generalize to unseen data. Therefore, while testing on natural stimuli is important, it is also critical to probe how the models generalize to examples that are qualitatively different from what the models were trained on. Some argue that we shouldn't expect neural networks to generalize to out-of-distribution data. I think that the validity of this counter-argument depends on how we conceive our neural network models. If they are just tools for curve fitting, then indeed, out-of-distribution generalization is not expected. But if we want to seriously consider these models as implementations of neuroscientific hypotheses, then we should definitely test their out-of-distribution generalization and strive towards models that correctly predict behavioral and neural responses in novel conditions. One way to design out-of-distribution challenges is to introduce handcrafted domain shifts, for example, as in the studies on robustness to image degradations. Another approach is to let the models guide us. By using optimization, we can navigate the vast space of possible stimuli, searching for inputs for which the models make non-trivial predictions. We already have several tools for designing such model-driven experiments, stress testing the models in different ways. We can generate superstimuli designed to maximize the predicted neural response. We can generate adversarial examples designed to make models err. We can generate metamers, which Jenelle Feather will discuss in detail later today. And we can generate controversial stimuli, which are stimuli designed to elicit distinct predictions in different models. In my experience, the moment we step out of the training distribution and test humans with model-driven synthetic stimuli, the limitations of the models are revealed. Such experiments uncover discrepancies between even the best models and human brain and behavior. Here are a couple of examples from our work on controversial stimuli. On the x-axis here, you can see model behavior correlations for NIST classifiers tested with plain handwritten digits. Once switching to synthetic controversial stimuli, we see that not all of the models are equal in their human consistency, and we uncover significant gaps between all of the models and the noise ceiling. Discriminative classifiers that worked really well with natural data failed to predict the human responses to the synthetic controversial stimuli. We saw a similar pattern when we tested natural models as predictors of human relative acceptability judgments. When we use randomly sampled natural sentence pairs, all models seem to perform quite well. Once we moved on to synthetic controversial sentence pairs, most of the models turned out to be really misaligned with the human judgment. This is an extension of controversial stimuli to RSA. We compared human judgments of phase similarity to 6 neural networks sharing an architecture but trained on different tasks. When testing randomly sampled faces, all models explain the human judgments with a similar level of accuracy. When we switched to controversial stimuli specifically designed for this task, we uncovered a clear hierarchy of the models. I invite you to visit Wenchen Go's poster later today, where she will demonstrate how this approach can be taken into the magnet in a human fMRI experiment. To summarize, when experimenting with synthetic model-driven stimuli, it becomes clear that the real limitation of current cognitive computational neuroscience is not the data, it's the models. Specifically, we still do not have models whose predictions veridically reach out beyond the model's training distribution. In order to accelerate our progress, I believe that we should pay more attention to our out-of-distribution challenges and to developing models that can succeed in these tests. Thank you very much.
Hello.
Yeah. So our next speaker is Kalanit Grill-Spector, who is a professor at Stanford University. Unfortunately, she couldn't join us today due to flight delays, but she'll be delivering her talk virtually. And she's going to argue that we need new models, particularly models that not only explain cortical function, but also cortical topography. And so you can take it away, Kalanit.
Hello.
We can all hear you.
Hi, everyone. Can you hear me?
Yeah, we can. Let me just unmute her.
Okay.
We can all hear you.
Okay. Hi, everyone. I'm sorry I can't be in person. I will describe work done in my lab, primarily by Don Fincy, in collaboration with Eshed Margalit, Dan Yamens, and Kendrick Kay. So in the context of the GAC, I will argue that we are limited by two main things. One, hypotheses that focus solely on function, and two, the types of algorithm that maps from models to the brain. And we are primarily interested in my lab to use deep neural networks to answer why questions about the brain. And today I'm going to ask one such question, and that is, why are there parallel processing stream in visual cortex? So a prominent feature of the visual system organization is the organization into anatomical and functionally distinct processing streams. And in humans, there are thought to be three processing streams, and the predominant theory that these streams have evolved to accomplish different visual tasks. The ventral stream is thought to be involved in object categorization and object recognition. The dorsal stream is thought to be involved in object detection and localization. And the lateral stream is thought to be involved in action recognition and processing dynamic stimuli more broadly. So if we want to use deep neural networks to test this theory, we might develop models that are trained to perform tasks hypothesized for each of these streams. So for example, we might train one model to perform object detection, another model to do action recognition, and a third model to do object categorization. And then what we were going to do is map these models to the brain with the predictions that units trained for detection will map to the dorsal streams, units trained for action detection would be in the lateral stream, and unit trained for object categorization will map into the ventral stream. However, crucially, to test this hypothesis, we need to have a one-to-one mapping between each model unit and a brain voxel. And this kind of mapping has not really been done in the field. So to do that, we developed a new algorithm that does a one-to-one matching between each model unit and the brain. And to do that, we use the natural scenes dataset, or NSD, in which we have brain responses for a rich set of images, thousands of images per voxels. And basically, we're going to obtain brain measurements and deep neural network measurements on the same images and do a one-to-one matching by computing first a cost function that looks at the pairwise distance between responses of each model unit and voxels for the same images, and then run an iterative minimization algorithm based on the Munkred's algorithm so that we find a unique match between each voxel and each model unit that minimizes this cost function. So how does this work out with our networks? I'm going to show you results on an example subject where the dots are colored by the tasks that they're trained on, and you can see that units trained for different tasks don't really cluster to a particular stream. And if we quantify the amount of voxels that are matched for each stream, we can see that performance is at chance. And even for an object categorization task that's been used to map the ventral stream, only about a third of the units trained on object categorization end up being mapped to the ventral stream. So maybe we need to explore an alternative hypothesis that task is not enough, and perhaps a set of anatomical constraints is necessary to explain this organization to streams. And put another way, perhaps visual cortex is optimized to balance learning useful representations with a spatial constraint that encourages nearby units to have similar responses. So testing this kind of hypothesis necessitates a new kind of model, specifically a topographic deep artificial neural network model, which I'm going to call a TDAN, in which both functional and spatial constraints can be implemented. So in a TDAN, prior to training, each model unit is assigned a retinotopic position and a 2D simulated cortical sheet for each layer of the network. And then during training, we use a loss function that includes both spatial and functional component, and the spatial component is weighted by a parameter alpha. And I'm going to show you results for a TDAN that was trained with a contrastive self-supervised training and an intermediate spatial weighting. So the top shows you the simulated cortical sheet of the last layer of the network, so where each dot is a unit, and they're colored by their location on the simulated cortex. And then we can look how they map into the brain. And you can see that units that are clustered in the simulated cortex are also clustered on the cortical sheet. And if we compare the proportion of units that are assigned to each stream, we can see that this is significantly better the chance, and for the ventral stream, we're approaching the noise ceiling. But perhaps this task optimized model better explains function than our TDAN. So we can compare the cross-validated functional similarity between each of the models and the cortex on left-out data. And we can see that for the task-optimized models, the functional match is far below the noise ceiling for all of the models in stream. In contrast, there's much better functional correspondence between the TDAN and cortex, where for the dorsal and the ventral stream, again, we're approaching the noise ceiling. We can also compare performance on the TDAN trained on different tasks, for example, the self-supervised task or the supervised categorization task, and also the functional spatial weighting. While we can see that there is an optimal spatial weighting, we can see that across weights and streams, the self-supervised task better explains the brain than the supervised categorization task. And crucially, in order to find the separation between models, a one-to-one mapping between units and voxels is necessary because the standard linear regression really does not separate these models. So to summarize, a topographic deep neural network with self-supervised training and a spatial loss function that encourages nearby units to have similar responses better explains the emergence of visual processing streams and task-optimized model. And direct unit to voxel mapping better separates models in linear regression. And finally, architectures matter a bit, but training loss and training set matter a lot. Thank you.
Thank you, Kalan. She's not allowed. Our next speaker is Katherine Hermann, who is a research scientist at Google DeepMind. And she's going to argue that for human-like models, we probably need to train networks on human-like tasks.
Okay, so in recent years, as has been mentioned, there's been a wealth of work demonstrating remarkable correspondences between models trained to categorize natural images and the primate visual system. And some of these papers are what got me interested in this field in the first place. But even so, as this audience knows, there's some salient mismatches between human and machine vision, especially when it comes to behavior. So for example, models are susceptible to adversarial examples. They rely on shortcut features like texture or image background, and they fail to handle common corruptions which don't foil human vision. So what would it take to close the gap? Plausibly, many of the divergences between models and people that exist now can be explained in the framework of shortcut learning. So when trained to categorize images, models may leverage so-called shortcut features, which are sufficient to solve the training task, but which don't generalize in expected or desired ways. And critically, for our purposes, they can differ from the ones that people use. I'm going to argue that most of the models that we study fall into this regime, and that the only reliable way to eliminate shortcut learning and bring models truly into alignment with people is to train models on tasks that capture the richness and variety of problems that our visual systems solve. So let me flesh out these points a little bit. In real-world objects, visual features like shape, color, texture, and background, just to name a few, are correlated to differing extents with object labels. So for zebras, both shape and stripes are diagnostic, for example. And in general, in naturalistic data sets, there are often multiple input features that predict task labels to differing degrees, and recent work has shown that people and models can make use of different ones. A hypothesis is that this could be a big part of what underlies differences in their behavior. So I'm going to spend the next few slides talking about one case study in differential feature use, because it's fairly well understood by now, and that's the differential use of shape and texture. So by now, you're probably all familiar with Robert Giros and colleagues' landmark study on texture bias in models, but I'll run through it briefly to refresh. So the authors presented ambiguous images like these, which are consistent with opposing shape and texture labels. The image on the left is the shape of a cat and the texture of an elephant. And people selected the shape labels nearly all the time, whereas CNNs trained to classify ImageNet images selected the texture labels the majority of the time. So when the field thinks about how to make more human-like models, there's a lot of attention to things like architecture, or training methodology, or moving to a contrastive objective from a normal supervised one. And here we have a nice setup to ask whether these sorts of manipulations actually do bring models and people closer. So with Simon Kornblith and Ting Chen, we found that they don't bring us quite there yet. To be clear, both architecture and training objective do influence a model's level of texture bias to an extent. On the architecture front, we know that CNNs and vision transformers vary in their particular level of texture bias, and also that transformers are more inclined to classify by shape than a ResNet counterpart. But all of these models still lag far behind human levels of shape preference. And at least so far, architecture doesn't seem to solve our problems. So perhaps none of these architectures are right, and we need to consider a broader class of architectures. And in particular, this would be true if these architectures classified by texture because they're unable to learn shape. We did an experiment to test this. So we trained randomly initialized models to classify these ambiguous images by either their shape or by their texture. So training on those tasks directly to see if the model could, in principle, learn each one. And we found that they could. So it wasn't the case that the models couldn't handle shape for these stimuli. But when trained on ImageNet, they end up preferring to classify them by texture. So what we've learned here is that even the simplest architectures can learn either of these features to the extent necessary to classify these stimuli, and it's training on ImageNet that seems to be what matters for texture bias emerging, a point that I'll return to in a minute. On the training objective front, some self-supervised training objectives reduce texture bias relative to the standard classification objective. But all of these objectives still produce a net preference for texture rather than shape. Dataset statistics, however, can really shift the level of texture bias around. So in particular, if we change up our inputs by applying data augmentations, we can either increase a lot or decrease a model's level of texture bias. And when we combine certain ones, we can induce an active preference for shape and better handle out-of-distribution images like sketches, where shape is important. So here we see the importance of what I've been calling task, although perhaps so far in a slightly contradictory way. So if we consider task to be the conjunction of the data that the model sees and what it's being trained to do with it, the importance of the data part here is already clear. For the objective, though, I'll argue that we should move much farther from the standard ImageNet training than we do when we try things like contrastive losses. What is it that people do with vision? So certainly we do categorize objects, but in some sense, most of what we do is interact with objects, figuring out how to sit in a chair, for example. And these interactive experiences are important during development. It should be intuitively obvious that these tasks just aren't solvable with texture-based shortcut features in the same way that ImageNet categorization seems to be empirically. So I'd bet that models trained to support physical interactions would have shape reliance that's much closer to that of humans than the models we have now. Zooming out and going back to my previous thread, texture bias is just one of many shortcuts that models seem to learn when tasks are solvable in multiple ways. So as a couple examples, models prefer to classify by local rather than global image content. So in a particularly extreme example by Gaurav Malhotra and colleagues, by the single diagnostic pixel in red rather than by any semantic content at all. As another example, image backgrounds rather than foreground objects can play an outsized role in driving model class label predictions. As an aside, plugging my recent research, an interesting question is why certain features become shortcuts for models when others are also predictive. And I'll just note in passing, this is something I've been investigating with my co-authors and would be happy to discuss offline, but label predictivity alone isn't sufficient to explain which features a model will learn. As with shape and texture, I think we can again be confident that these other kinds of shortcuts would be much less likely to emerge in models trained on ecologically realistic object interaction tasks. And overall, if we want to make models that are more like humans in their behavior, and we have some fixed budget of time or resources, my claim is that we should spend most of it on creating ecologically realistic training environments. Behaviors and representations for both models and people emerge to solve the problems they're faced with. So if we want similar behaviors, we need similar problems. But what might this look like concretely? In a commentary with Aran, as well as Sjoerd van Steenkiste and Matt Jones, we argued that one promising direction is to situate a vision system within a simulated agent who's navigating and interacting with its environment. But to do this well, we need to continue to develop good naturalistic training environments with sufficiently challenging possible tasks. We also have language models now, which we can use to communicate richer tasks to models, and which can also provide a reservoir of human-like knowledge for models to draw on. Now, of course, this enterprise is going to take time. It took a great deal of work to create data sets like ImageNet, and the tasks I'm arguing for here are much more complex. But I think this is what we have to do if we want to reliably create models that match human behavior in the ways that we care about. Thanks.
Hi, so now we'll have the audience Q&A. So we're actually running a little bit late, so we'll have to cut short the Q&A. We can probably have about 5 minutes of Q&A. And we already have a couple of highly upvoted questions on Slido, so probably we can just start with those. So here's a question by Dan Goodman, who basically asks, how can we test null hypotheses that any model that extracts relevant statistics of the environment will have some correlation with brain state? I think this could also be a good question for the tools and metrics section of the workshop. Maybe we can also bring it up then. But would any of you volunteer to share your thoughts on this? I guess another way of kind of rephrasing it would be, do we have negative examples of models, for example, that are trained on natural input statistics but really do fail to show correspondence based on any measure like correlation with brain state?
You're saying if I train a model on a naturalistic data set, does it fail to match neural dynamics or something like that?
Yeah.
Okay. Yeah. So, I mean, in the mental Pong data I showed, this was the case with ImageNet optimized models, CNNs, transformers of the sort, and even when they were equipped with dynamics.
One more example is the first generation of autoencoders. So even if you train them on ImageNet, they don't align well with brain data.
I guess we have another question that's highly upvoted. So I don't know if we have Kalanit over Zoom. Maybe we can just skip that question. Then the next question is, what does out of distribution mean for a large language model that has been trained on billions of text documents on the web? Maybe Tal, do you want to take a stab at this question? Since we talked about out of distribution stimuli.
So we've been playing with language models, not large language models yet, but they do have a distribution and out of distribution, right? There's the distribution of English or other languages on which they were trained, and there are many sequences of potential combinations of words. And we would like to be able to predict how humans respond to such sequences well. I don't think that we are there yet.
Then we also have another question which is very pertinent to this workshop. So the question goes, is comparing artificial and biological networks even the right approach? Has it provided any truly novel insights into brain function? Would anybody want to answer this question? I guess the question is what have we learned from all of this whole comparative analysis enterprise thus far? Or what do you think is the most interesting thing that we have learned from this whole research endeavor?
I mean, I think there's several things we've learned, but I guess I'll maybe mention one that stands out the most is, I think for a long time in systems neuroscience, there were tons of neural responses that were just very hard to ascribe a particular function to. They just weren't really nicely written down in natural language. And we built handcrafted models to explain them, and we could explain very small fractions of these populations. So to give an example, like grid cells in medial entorhinal cortex, very simple cells in V1, et cetera. So, but now, I mean, I think at least in the domain of vision, we now have a way to basically have a handle on these other kind of messier, more heterogeneous cell types by way of functional optimization on a behaviorally relevant goal. And I think we're also starting to see that pay out in higher cognitive areas, not just in hippocampus, but also in frontal cortical areas, where actually that data is starting to push down a lot of the current models we have and suggests strong constraints. For example, that these reusability is a very important condition, and having things like object slots or pixel-wise predictors, or even reinforcement learning end-to-end seems to be a poor idea for matching both visual cortex, but also more sensory-aligned cognitive areas like dorsomedial frontal cortex. And I think a curious observation, I guess, that I've seen both in rodents and in primates is that, just to the last point, is not only did we see the failure of end-to-end reinforcement learning in this data, but also even in rodent visual cortex, when we train agents in naturalistic environments end-to-end with reinforcement learning, it seems to fail compared to more generic sort of contrastive self-supervised methods. That's not to say that they've solved everything, but it's just a suggestion that perhaps there's some type of modularization that might be going on, whereby sensory systems have to solve a more generic goal, and maybe reinforcement learning could take a role in motor cortex or other areas more specifically. And that's just a last kind of note on my end, at least.
And then we have one question that's particularly directed to Kalanit. So the question is, why expect a one-to-one mapping between ANN and brain units when the brain units in question are fMRI voxels that aggregate over thousands of neurons activity?
Hi, this is a very good question, and I have thought about it a lot myself. So I have several answers for you. First, I think we're past the idea that there are grandmother units in the brain, and probably the computational unit in the brain is a population of neurons rather than a single neuron. And if we think about the point spread function in the brain, a cortical column is about, in human brains, about 1 to 2 millimeters, which is in the sense, the scale of MRI voxel, especially in the NSD data. The second thing, if you think about microstimulation experiments, in order to disrupt behavior, you really have to stimulate a population of neurons. Again, not a single neuron would cause some behavioral deficits. And finally, I'd like to point out that even, and this is back to the question, what have we learned about these models about biology? We have to remember that our model units are in some way an abstraction, and I don't think that we can really make the inference, direct inference that a model unit is equivalent to a neuron. Nonetheless, what the method that I've described them, Vox is a model to brain mapping, the direct mapping, it depends on the data type. And if you have electrophysiological data, that you have single neuron data, you can apply the same kind of methodology to single neurons.
Thanks, Kalanit. And just one last question. Do we have time for one last question?
We'll take it over Slido.
Okay, we'll just take it over Slido, or if you have any questions, you can also discuss those with the team members after the workshop or during the break. Sorry, we are really time constrained. So we'll get started with the next section of our workshop, which is on tools and metrics. Thank you. So, thank you to all the speakers for this session.
Okay, I want to add one more thing, which is that the audience members should keep asking questions, because we can still answer all of those questions on Slido directly. We will share all the questions with our speakers, and they will be able to do that. So with that, I would like to actually invite the next group, which will be talking about metrics. Our first speaker is Meenakshi Khosla, who is a postdoctoral scientist at MIT, working with Nancy Canvisher.
Hello, everyone. So the title of my talk is Privileged Representational Axes in Biological and Artificial Neural Networks. And my view is more strongly conformed with the idea that we are currently limited by tools and metrics. And so what's missing? The TLDR version of this talk, and basically my answer to this question, is that one of the limitations of our current tools and metrics is that they ignore representational axes. And so let me first expand on what I mean by that. So in order to understand how neural systems process information from our external environment, two frameworks or two viewpoints have been commonly proposed in the neuroscience community. Under framework 1, how individual neurons implement neural representations is often considered irrelevant. So to explain what I mean by that, let's consider the representational system that's illustrated in this schematic system A. So here we have the cardinal axis representing individual neurons, and different stimuli produce different patterns of neural activity. And so now imagine that we can arbitrarily rotate the coordinate axes of this representational system, and it gives us a new system. Let's call this system B. And since we only rotated the representational axes, the relative distances between stimuli in this new representational system are also identical to those in system A. And similarly, what we can linearly decode from this system is again identical to system A because we just changed the bases. And so viewpoint 1 basically says that we should treat system A and system B as equivalent systems. Viewpoint 2, on the other hand, says that what individual neurons encode holds relevance. And so note that when we rotate the coordinate axes, we change the tuning properties of individual neurons in the representation. And so viewpoint 2 basically says that the representational coordinate system matters and that we should be treating system A and system B as different systems. So how do we decide which framework is more suitable for understanding the neural code? Note that there are really two questions that are relevant to choosing between these two viewpoints. So one is an empirical question of whether the brain does indeed have a preferred way of assigning representations to individual neurons. And the second is a theoretical question of why should a brain region even care about how its representation is implemented at the neuron level. And we currently don't have strong quantitative evidence or a theoretical clarity on these questions. And moreover, the tools that we employ to understand the neural code often implicitly align with viewpoint 1. So they assume that the coordinate system should be treated as arbitrary. So for example, one popular approach to understand neural computations is to compare the alignment between representations in brains and artificial neural networks. And the focus so far has largely been on comparing representations at the population level. And all the popular current tools like encoding analyses, RSA, or centered kernel alignment, they all come with an implicit assumption. And the assumption is that they're all invariant to axis rotation. So a representation R is treated equivalently as a transformed representation R prime, which is obtained by rotating the original representation's axes. And so these methods implicitly assume that the representational axes are irrelevant. So they're sensitive to what can be called the representational content. That is what information is linearly accessible from a representation or its geometry. But they're insensitive to the form of the representation, so which is how is information represented in individual neurons of the representation. And so by disregarding the explanation of this representational form, I'd argue that important data remains unexplained. So coming back to the question that I posed earlier, how do we answer whether brains have privileged bases, and what are its computational consequences, if any? So in recent work, we propose a formal framework for assessing whether and to what extent a representational system has privileged axes. So the idea is quite simple. So assume that we have representations from two different instances of a system. Let's say we have representations from two different subjects to the same stimuli. As part of step 1, we can compute the alignment between these two representations. And now we can randomly rotate the axes of representation in one of the instances, so in one of the subjects, let's say, and then recompute the alignment between the representation with the rotated axes and the original representation in instance 2. And so more formally within this framework, we draw a random rotation matrix uniformly from the set of all possible rotation matrices. And then we draw smaller rotation matrices by computing fractional powers of this rotation matrix. And then for each of these rotation matrices, we can then compute the alignment between the original representation in instance 1 and the rotated representation in system instance 2. But we still have to choose an alignment metric in this framework. And like I said, the existing metrics that we've been using so far, they're all invariant to the representational axes. So we cannot employ them to ask this question. So we need new metrics that are rotation sensitive. And so for that, we propose novel axis-dependent alignment measures. So for example, one intuitive metric that we use quantifies the one-to-one alignment between units in two representations. And the second measure, which is theoretically much more principled and based on optimal transport, can be interpreted as a soft permutation between the units in two representations. We can also go one step further and run an optimization algorithm to find the optimal rotation matrix that would maximize this rotation-sensitive alignment between two representations and use that to compute the maximum possible alignment between any two representations under these bases, coordinate axis shifts. And so just to intuit what we should expect to see if a representational space indeed has a privileged axis. So if, for example, different branes have a preferred basis, we would expect the alignment between different branes to drop off with the rotation of the representational axes, and the alignment in the natural basis to be close to the optimal. But if instead there is no preferred basis, then we might see something like the pattern on the right. And so let me first address the question of whether branes do have a privileged representational axis or not. And so I'll first focus on the domain of vision. So here we wanted to know whether visual cortical representations have a privileged basis or not. And so to get at this, we ask, how does alignment between the visual cortex in two different subjects change as we rotate the representational axes in one of the subjects? And so in humans, we use data from the natural scenes datasets, where 8 subjects watched a common pool of 1,000 images. We compare the alignment between the ventral visual stream responses in pairs of subjects, and we study how this alignment changes as we rotate the representational axes in one of the subjects. And so here are the results. You can see that it falls off quite sharply with axis rotations, leading to an almost 25% drop in alignment with the full random rotation of the axes. And the alignment in the standard basis, which is the natural basis, is also close to the optimal. We observe very similar results with single neuron recordings in macaque IT. And so it basically suggests that the visual cortex does indeed have a privileged basis because its representations are most aligned in its natural basis compared to any arbitrary basis. We can follow the same approach in the domain of addition, and again, we find that the auditory cortex also has a privileged axis. One way, so sensory systems do seem to have a privileged axis, and one way to make sense of these privileged axes is to ask where and when parallel computational models of these areas also develop privileged axes. And so here we ask, how do arbitrary rotations affect the axis-dependent alignment between different deep neural networks? And so when we compare the alignment between deep neural networks trained with different architectures on ImageNet, so for example, ResNet versus AlexNet, a similar trend is observed that, that axis rotations consistently reduce the alignment. And we see the same trend when we compare networks trained with different objectives, or even networks that are trained with both different objectives and architectures. So this basically suggests that learning in different deep neural networks converges to a privileged basis that is shared across diverse architectures and learning objectives. And we observe the same trend in the domain of audition. And so this also suggests that deep neural networks do indeed have a privileged basis. So far I showed you that brains have a privileged basis, and independently neural networks also have a privileged basis. We can also ask whether they also have an aligned basis, or aligned representational axis or not. And we also have some evidence which suggests that that is indeed the case. So ANNs and brains are actually most aligned in their natural basis compared to an arbitrary basis, which strikingly suggests that they also share these privileged representational axes. And I won't go into too many details, but we also show in this work that there are important implications of this representational axes. So one is that they reduce downstream wiring costs. The privileged basis also promotes higher lifetime sparseness, and it can even facilitate few-shot learning under biological constraints like sparsity. And so just to kind of really conclude here, we showed that it is an empirical fact that brains and deep neural networks have a privileged basis. They often even have aligned basis dimensions. And so if the goal, and these privileged bases afford distinct computational advantages. So if the goal of the comparative analysis framework is to understand the neural code from a functional and a mechanistic perspective, then the tools that we employ perhaps should also be sensitive to this representational axis. Thank you.
Okay, so just a reminder, everybody, if you can ask your questions on Slido, and our code is GAC 2023. Our next speaker will be Alex Williams, who is an investigator at the Flatiron Institute and a professor at NYU.
Do you mind grabbing this, actually? Okay, great. It's cool to be here. I'm going to give a really short talk that just focuses on some limitations that I think are important about interpreting linear predictivity scores, and I'll get to a little bit of our research at the very end. So I know this is meant to be a provocative and adversarial session, and the predicament that I'm in is that I think I might have some adversarial opinions, but I'm not a very adversarial person. So I wanted to very briefly give a couple disclaimers at the outset. The first has already been mentioned. Do we need better metrics, better data, or better hypotheses? And obviously we need all of these things. The reason that I'm speaking under the metrics is, well, collecting more and better data is difficult, and coming up with new scientific hypotheses is arguably even harder. But I think coming up with metrics that we really understand the mathematical properties of is an extremely tractable problem. And my optimistic take is that over the next few years, I think we'll have a better understanding of the metrics we use, and maybe even come together as a field to decide, what are the best metrics for each situation. Other quick disclaimers. For the purposes of this talk, I'm going to kind of create an oversimplified straw man argument that I call the linear predictivity hypothesis. This is not meant to be an attack on any individual's point of view or work. The idea is that I think even though this is potentially a straw man argument, it's a useful one for us to kind of consider together and talk through potentially some of the issues. Okay, so what do I mean by this linear predictivity hypothesis? So this is an argument that basically says if I have two models, x1 and x2, and some brain data y, I'm going to compute the percent variance explained in y by each of these models, and I'll call that an R-squared score. Typically, you're going to measure this by some sort of linear regression model, often with regularization. So the main claim of this hypothesis is that if the R-squared value of x1 is greater than the R-squared value of x2, in terms of x1 explains more variance in the brain data, then that implies that x1 better captures the computational mechanisms in y relative to the other model. And this is the, I think, strong claim that I want to push back against a little bit today. Okay, so just to be clear about what my position actually is, I'm not saying that R-squared values are useless and that we shouldn't look at them for certain things. In fact, it's arguably maybe a good measure if you're interested in running in silico experiments where you present, for example, stimuli, images to deep networks, and try to predict how biological neurons are going to perform on those unseen images, although we've seen that there's limitations for out-of-sample generalization. So predictivity scores are useful for certain things, maybe visual prosthetics is another one. But my argument is that predictivity scores should not be interpreted as a measure of model similarity. And the simplest way that I think I can show you this is with the following one-dimensional example. So just think about tuning curves to a one-dimensional stimulus like oriented filters, like classic Hubel and Wiesel visual stimulus. I want you to think about two systems now, system X and system Y. And the important thing is that the tuning curves in system X are uniformly tiling the stimulus space, but the tuning curves in Y are completely blind to 50% of the orientations, if you will. And hopefully, I think everybody in the room can agree that these are fundamentally different systems. If there's downstream computations happening, they're likely to be different. For example, system X contains fundamentally more information than system Y. There's fundamentally information missing in Y. Nonetheless, you can fully reconstruct the tuning curves in Y to high precision with a linear model using the tuning curves in X. So the R-squared score is essentially 1. And if Y were to represent the brain and X were to represent a deep network, the linear predictivity hypothesis would falsely conclude that X is a perfect model of Y. Another way to kind of see the faulty logic here is to see what happens when you swap the independent and dependent variables. So you can predict Y from X perfectly, but you can only very poorly predict X from Y. And the problem I see is that a lot of papers in our field only are reporting R-squared scores in one particular direction and not providing this bigger picture. Very quickly, in preparation for this talk, I was reading a nice set of papers by Rosa and Dan Yeamans, and they make a lot of interesting points in those papers, but I think one of them is pretty similar to the argument I just showed you. So I'm not going to read this whole thing, but very quickly I'm going to say that they cite the universal approximation theorem, which roughly states that any function generated by a deep multi-layer neural network can be approximated by a shallow network with a single, potentially very large hidden layer. And importantly, they conclude that this does not mean these two networks are similar to each other because it fails what they call the mappability criterion. And at the risk of misinterpreting or oversimplifying their argument, I think what they're getting at is something very similar to what I said before, namely that a very wide and shallow neural network can approximate the function of a deep neural network, but somehow the linear mapping from left to right on this slide is fundamentally different from the linear mapping from right to left. And in fact, in the setting, I think that you can see that the R² score can be nearly equal to 1 in one direction and nearly equal to 0 in the other direction, in the limit that the width of this layer becomes infinitely wide. So this leads me to ask, what do we actually want what do we want out of a metric if we want to quantify similarity in neural representations or neural computations? I think what linear predictivity gets right is that it wants a measure that is, or a distance measure that's equal to 0 when there exists some simple transform f within a class of mappable transforms that maps x onto y. And this is essentially what Rosa and Dan also argue, and I think Aran in his talk brought up very briefly. But I would argue that we need additional requirements on our metrics. I would advocate for the notion that the distance measure should be symmetric, so the distance from x to y should be the same as the distance from y to x. RSA and CKA are metrics that satisfy this condition. And then finally, I would also argue that you want these distance scores to be self-consistent among not just pairs of networks, but 3 or more networks. And the self-consistency condition that mathematicians and data scientists, machine learning people really like to see is the so-called triangle inequality. And I can go into the details about why I think this is important. But the summary of my talk is that R² scores or linear predictivity scores fundamentally don't satisfy the second and third axioms here. But there is really no need for us to compromise on this. We can come up with distance measures between neural systems that satisfy all 3 of these and furthermore generalize to different function classes of mappable transforms that we want to quotient out or consider equivalent. And I don't have all the time to go into the details of that, so I'm going to point you to these 2 papers from my lab, and there's other work coming out. Our code associated with these metrics is also available on GitHub. Our ideas are measuring well-known ideas from a mathematical field called shape analysis or shape theory. Very briefly, what this lets us do is compute distances between 2 networks that can be interpreted, in certain cases at least, as a geodesic distance on a manifold of possible network representations. And we can leverage that to do really interesting things like find clusters of neural networks that have similar representations to each other, and even do supervised analyses, so like predict what a network's test performance would be just by looking at its hidden layer representations. This is what it looks like on real biological data. I don't have time to explain that. I'm already over time, so I'll just thank my lab quickly and pass the baton on.
Okay, so our next talk is by Jessica Thompson, who is from here, the University of Oxford.
Okay, great. Good morning. I'm Jess, and my position is that tools and metrics are means, not ends. This GAC asks whether our scientific progress is limited by tools, data, or hypotheses, but in pursuit of what goal? What goal were we moving towards before that we are now stalled in our progress toward? We have to know where we're trying to get to before we can know what's blocking us from getting there. Sometimes this discussion is framed as if the goal is model improvement or model refinement, that we are gradually narrowing in on a more brain-like model until we someday understand the brain. I'm certainly guilty of this kind of thinking myself, but I think it gets us into trouble because it makes the brain to model comparisons an end in themselves rather than the means of answering some scientific question. So rather than starting with the model and asking, how do I make this model more brain-like, let us start with the behavior and the experience to be explained. What is the specific question to be addressed? Then neural networks provide a framework in which to formalize our theories. Importantly, we can't expect to explain everything at once. The relevant explanatory factors for one phenomenon will be irrelevant for another. You have to choose. In order to develop the right tools and know when to deploy one or another, I argue that, as a community, we need to spend more time critically evaluating what questions we actually want to address. What are the questions that are worth asking that are also answerable? And how will our model-to-brain comparisons serve that scientific project? Let me give some examples to illustrate. Here I'm going to contrast two papers that make opposite assumptions about what counts as a meaningful difference between two representations. This paper assessed the similarity of representations in identical networks differing only in their random initialization. They found that while early layers tended to be very consistent, that there was a lot more variability in the later layers. And then they concluded that practitioners should always repeat their experiments with several random seeds in light of this. This is presented as an empirical result, but contained within it is also a value judgment that this difference in representation is meaningful for neuroscience. That judgment is supported by the fact that in assessing similarity, they used metrics that are commonly used in systems neuroscience, suggesting that the outcome of a particular analysis involving deep neural networks could depend on the particular random seed employed. Contrast that with this paper, which starts by assuming exactly the opposite, that the representational differences arising from different random initializations are not meaningful, in this case for machine learning researchers seeking to understand deep neural networks. They start with a desired qualitative notion of representational correspondence, namely that a good measure of similarity should find corresponding layers in multiple instances to be most similar. So layer 1 of instance A should be most similar to layer 1 of instance B. Layer 2 of instance A should be most similar to layer 2 of instance B, and so on, yielding high similarity along the diagonal of this similarity matrix. From this, they conclude that measures based on centered kernel alignment are more suitable for this purpose than those based on linear regression or canonical correlation analysis. So here we have two pieces of research that, because of their different use cases, have made exactly opposite judgments about what kind of representational correspondence is meaningful. As another example, consider this hypothetical scenario. Say you want to understand the primate visual system, and you notice that the dominant neural network models only consider the ventral stream. So you figure a better model of the primate visual system should include the dorsal stream. So you go about building a dual stream network to mimic the parallel pathways of the primate visual system. Now you want to evaluate the model, so maybe you collect some new brain data and compare the biological neural representations to the model activity. But what if you find that actually a simple CNN explains just as much variance in the data as your new dual stream model? What does this mean? What do you do? Probably you should organize a GAC about it. But alternatively, you might approach this completely different from the beginning. You might start by asking, why did we evolve to have two visual pathways in the first place? Why do the two streams have the particular functional specificities that they do? What cognitive ability does this structure serve? You might then start to think about the role that the dorsal stream plays in understanding the relational structure of visual scenes. You might then build a dual stream neural network model that embodies your hypotheses about the particular aspects of the primate visual system that are relevant to this capacity. You might then evaluate the model by showing that it replicates various neural and behavioral signatures of the cognitive phenomena to be explained without ever making any new neural measurements or even doing any quantitative model-to-brain comparisons. And in fact, this is what we've done, and you can hear more about it tomorrow at our poster. So in summary, any model-to-brain comparisons are not the end in themselves. They serve some other scientific goal of describing, explaining, or understanding some perceptual, cognitive, or neural phenomenon. Which comparisons will be useful will depend on the specific scientific question to be addressed. We are limited by tools and metrics because we fail to reason about what evaluations of our models are meaningful in light of those specific questions. We're not limited by data because we've already collected so much. There's so much that we already know about the structure and organization of the brain that begs for explanation. And this is where our neural network modeling can come in. And we're not limited by hypotheses, but because there's so much yet to be done within the machine learning frameworks that we have. The frameworks are general enough that new hypotheses can be incorporated easily. And please forgive all the caricatures. And that's it. Thank you very much.
And our final speaker for this session is Bradley Love, who's from the University College London.
Great, thank you. So what do neuroscientists usually mean when they say a network layer corresponds with the brain region? Usually what they mean is that there's a correlation between model and brain activity. So for example, in encoding approaches, you could take some activity in a model layer and via linear regression account for variance in some brain region. Likewise, in representational similarity analysis, you have a pattern of similarity on the model side, a pattern of similarity on the brain side, and you correlate them. And other approaches, from canonical correlation analysis to center codon alignment, have this similar underlying logic, namely that correlation implies correspondence. And using that logic, the field settled on this interpretation of the ventral visual stream for object recognition, in which there's this early to early, late to late pattern of correspondence between model layers and brain regions. So for example, an early model layer might correspond to V1 through these correlative approaches, and a late model layer might correspond to IT, a late region along the ventral visual stream. So early to early, late to late. But does correlation really imply correspondence? Because when you think of the brain, a lot of the activity, a lot of the variance going on might not even be related to the task of interest. So really we only care about the variance, I think, that drives the behavior, the task-relevant variance, if you will. So I'm going to propose a stricter test of correspondence. It's a direct interface or substitution criterion. So namely, if a brain region really corresponds to a model layer, then you should be able to replace it. So you should be able to take the model activity, replace it with brain activity, and drive the computation forward towards some outcome, towards an output, towards a behavior. And so that way you could really say, this model layer is playing the same role in the overall computation as this brain region. So we're going to do that. And the idea here is normally in object recognition networks, you follow this blue pathway. You show the image to the model, and you get an output. So hopefully in this case, ostrich along that blue pathway. What I'm saying instead is if some brain region corresponds to some model layer, then instead of showing the image to the model, we could show the image to the brain, take that brain activity, and through a simple learned linear translation from brain space to model space, we could substitute the model activity with the brain activity along this red pathway and drive the computation forward. And hopefully, if there's a good correspondence along the red pathway, we'll get ostrich as well. So this is work done by Nick Sexton, who's great. And so we're just going to do this. First, we're going to look at an adult fMRI data set in which human adults looked at photographs of natural images. And so what I'm showing you here is the performance of the model when we interface it with brain data from lateral occipital cortex, LOC, which is a late region along the ventral visual stream. All these bars here correspond to different model layers that interface with that brain data. From left to right, it's from early layers to late layers. So you can see here, by using this direct interface approach, substitution test, we get a late-to-late pattern of correspondence. The model performs best when LOC data is interfaced with the later model layers. However, what happens when we interface brain data from an early visual region? We don't see early to early. We see that these regions look functionally like late model layers as well. So the model performs best when these early visual regions are interfaced with late model layers, just like LOC. And same goes for these other brain regions. So that's just one study. Let's look at another fMRI study of adults viewing photographs. And here's LOC again, which is a late region along the ventral visual stream. And once again, we see this late-to-late pattern of correspondence, which is consistent with the zeitgeist of the field. But however, if we look at these earlier visual regions, V1 through V4, we see the same thing. We don't see this early-to-early pattern of correspondence. Functionally, all these regions look like late model layers, as does FFA and PPA. So maybe fMRI, it's just problematic, and let's look at some monkey data, some multi-unit data from monkeys, looking at these odd images. So we had to make one minor alteration to the method here, because these images do not correspond to standard ImageNet classes. But we just showed these images to the model, like along, say, the blue pathway. We get an output for each one, like a 1,000-dimensional probability distribution of the things the model can say. Then we did the same across the red pathway with this brain-machine interface approach, and we get another 1,000-dimensional probability distribution. And we just want them to be the same, so now we're going to use a distance measure, KL divergence. So now lower is better. Okay, so if we look at IT on the right up here, this is a late region along the ventral visual stream. We could see we get a better match to late model layers. This is late to late, so now lower is better, it's distance. But if we look at V4, which is an intermediate region along the ventral visual stream, once again, just like the fMRI data, we're seeing that functionally it looks like a late model layer. So what's going on here? So across all these studies, nothing looks like an early model layer. Everything looks like a late model layer functionally, like the role it's playing in the computation. And so we had this hypothesis that there's long-range recurrence going on. So after 60, 70 milliseconds, basically there isn't an early model layer, functionally, early brain region functionally. So I think this is a case where, and I should say we did some time series analysis with the monkey data to support that hypothesis. So I think this is a case where looking at shared or common variants could be misleading, because these early regions along the ventral visual stream, they can be dominated in terms of their overall activity patterns by stimulus properties, but as the computation unfolds, their role in the computation changes, and functionally they're really behaving like a late model layer, even if very little of the variance there is reflecting that. Okay, so this is not to say that, just to assuage some fears that we always find late model layer correspondence with this technique. We made some synthetic data. We took some images, so not brain data, where there's no recurrence going on, and just did a PCA of them. So it's very image-like, and then we just did this approach, and now we do find an early-to-early correspondence. So this technique can find early-to-early correspondences when it's appropriate. And finally, just to say something nice about these networks, so it's not the case that they're without value. So they're picking up on constraints of human vision just by virtue of being trained on like million naturalistic images. So these are some zero-shot decoding results where we took this red pathway and we held out whole object classes, say like we didn't ever train it on ostriches. And if you showed an ostrich for the first time, it turns out this translation on the red pathway from brain space to model space is fairly general, so you could project into this model space and the model will be fairly accurate in doing this like zero-shot decoding and saying ostrich. Okay, so in conclusion, this direct substitution, direct interface approach finds everything in the brain is functionally, a late model layer. So there's not early to early, late to late. And one explanation that we supported in some time series analysis that I didn't have time to show you is that there's long-range recurrence, even 70 milliseconds in. There's this tinge of like this signal from higher level areas like IT. And so even if most of the variance, in a brain region is reflecting the stimulus properties, its role in the computation is more, advanced at that point in processing. So what does it mean for a model layer and a brain region to correspond? Well, I think this is something we should reflect on and make explicit. For me, it means that they play the same role in the computation, so you could be able to substitute one with the other, and you could trace that out to an output, to behavior ideally. Yeah. So I'll stop there, and thank you all.
Okay, so with that, we will conclude this session. Unfortunately, we will not have time for questions because we are already eating into your tea break. So join us again at 10:45. I made one mistake. The next session will start with Rosa Cao's talk, and then we'll move on to the other talks. Thank you. We're going to start exactly at 10:45.
Hello, everyone. We're going to be starting soon with the next session, so I would really request you all to settle down. So we have one more speaker from the last section on tools and metrics. So we have Rosa Cao who's a professor at Stanford University, and she's going to be talking. The title of her talk is looking at similarity from the brain's own perspective, and so I'll just give it.
Everyone got revivified from the coffee. So I'm really lucky to go after all these great talks from the last session with really great examples, because now I don't have to present any. And actually, like, the punchline of my talk is in the title. So I think we should look at similarity from the brain's own perspective. But to make that case, I'm going to back up a little bit. I'm a philosopher, and I've been thinking about the concept of similarity and how it's useful in science from sort of a more theoretical perspective. And I'm hoping that that perspective tells us something about what kinds of specific metrics, what kind of strategy we should use for generating specific metrics for comparing models and brains. Okay, so whether neural networks can help us understand how the brain contributes to our capacities depends on whether they're relevantly like us for the purposes of explaining these capacities. And we're interested in a quantitative measure of similarity. And when I think about why we should care about similarity for the purposes of science, there's this sort of cartoon version of how model-based science works, where you kind of make a strategic choice. You can either study a system directly, you can do traditional neuroscience and stick electrodes in the brain and image it and see what it's doing and see what animals are doing, or you can take a strategic step back and try to, instead of identifying and describing the actual system's parts and their workings, you deliberately describe a different system, a simpler hypothetical system, and try to understand that other system's workings first. And I take it that that's the approach that we're trying to take by looking at neural network models of the brain. And in this diagram, the model system is the hypotheses. The target system generates the data. It's not actually exactly the same as the data. We make some other choices about what kinds of things we want to measure from the target system that we think are functionally relevant. And this resemblance relation is supposed to be captured by the metrics or the tools that we use to compare the two. But the idea of similarity is pretty fraught. What does it really mean to say that a model system resembles its target? There's this sort of old canard that everything is similar to everything else in some respects and not others. And so any kind of comparison is maybe not even informative. So Quine famously said that similarity is he said something like, it's logically repugnant. Right? So there are these issues with similarity. Here's an old example from Nelson Goodman highlighting the context sensitivity of similarity judgments. It seems like, depending on what your interests are, whether you care about how heavy something is or what color something is, you're going to make quite different similarity judgments. And Goodman's conclusion from this is that any judgment of similarity is vacuous and reflects only our prejudices and doesn't really inform us about the world. So that's a real worry if we want to employ similarity in scientific discovery and in model-based science, right? We don't want it to be a subjective measure. We want there to be some ground truth about whether things are actually similar or not. And there's a question about how we really assess similarity. And that's a problem that shows up in practice and not just in theory, as we've seen. I won't go over this. You've seen it multiple times by now. But we want to know, the breakdown of the correspondence between task performance and predictivity, you might think, is a kind of symptom where there are networks that are similar in some respects and different in other respects. And it's an open question whether they're different in functionally relevant respects, or if they're different in origin and construction, like the visual transformers and the MLP mixers and the CompNets, but maybe they're actually functionally similar to each other once they're trained. So it's not really clear whether a similarity measure that pulls them apart is capturing functionally relevant differences or not, depending on what task or what cognitive capacity you're interested in explaining. Okay, so given that there's all this context sensitivity, how do we decide on which features matter? And how do we decide in a non-subjective way? I don't think there's one answer to that, but I think there's one kind of general strategy. And more generally, when you commit to an assessment, when you commit to a measure or a metric of functional similarity, that's a way of making concrete which properties you think are functional and important for the operation of the system, and which properties are important for understanding the operation of the system. Okay, so what are some candidate ground truths that we think a similarity assessment ought to capture? So here are some things that people have said, right? We should define the ceiling based on animal-to-animal similarity. Two models with the same architecture and training but with different initializations. Some people think that those should count as highly similar, maybe maximally similar for our explanatory purposes, and some people think that they should be different. That came up in the last talk. It does seem that everyone agrees that differences in behavior should be reflected in differences in internal representational similarity because something is generating that behavior. And whatever's generating that behavior had better be functionally relevant. Okay, so I'm not committed to any of these ground truths or candidate ground truths as listed here, but they do illustrate that similarity measures are constructed to reflect our priors about which differences matter and which ones don't. And specifying that measure makes particular things explicit. So here's a list from this other paper that's been mentioned a bunch, the Kornbluth et al., where there are lots of different possible ways that you could try to assess the similarity of neural network representations. And different people have answered it in different ways, especially Alex's work and this work from Jacob Steinhardt's lab. And the mapping is very commonly some kind of linear projection, but as we've heard, there are lots of other options. There might be something more restrictive, like Meenakshi's one-to-one mapping, or maybe something more complex, like a nonlinear mapping, something more restrictive. Okay, so that's a general shape of the problem. We've all already seen it. And I think this question about finding the best similarity measure is interestingly connected to another old debate in cognitive neuroscience and machine learning. And that question is, what are legitimate and illegitimate ways to extract representations from brain activation? So some of you might recognize this picture from the bad old days of early fMRI, where people were quite worried about the kinds of statistics that were being done to extract regions of activation. And so this is a fMRI of a dead fish, and they found functionally significant activation because they had overpowered statistical methods for trying to figure out which regions were active. So that's like an obviously illegitimate way. There are similar criticisms that are leveled at techniques like GWAS in genetic analysis. And then there are like more, quote, legitimate ways to extract representations from brain activation. So again, linear probing is a way that lots of people think is a baseline plausible way of extracting real representations from the brain. So the kinds of significant activations that are really there, and we're not just making them up because we have overpowered statistics. Okay, so I've argued that the kinds of probes that we use to extract data from the brain, the kinds of decoders that we use to try to decode representations from the brain are quite similar, and both of them should be brain-like, because in order for them to serve their explanatory function, they have to be sensitive to the same features that downstream parts of the brain are sensitive to, right? So that kind of makes sense. You want to capture just those parts of brain activity that are actually being used by downstream parts of the brain to do whatever interesting thing it is that it's doing. And I think the exact same thing should be said here for similarity mappings, right? Here's the analogy. We want to capture the ways in which a target and a source, or a model and a target, two different systems are really similar while still being sensitive to the ways in which they're really different. And if we know how much the model is different, we can change aspects of it to try to make it more similar, and we learn which aspects of the model actually matter for predictive accuracy. So what a similarity assessment should be sensitive to are, again, just those properties of the thing being compared that downstream parts of the brain should also be sensitive to. So this is what I mean by similarity from the brain's perspective. And Bradley Love's talk earlier was a great example of, I think, this kind of thinking, although I reach slightly different conclusions than he does. So, what's the best way of comparing the similarity of corresponding layers in a biological system and an artificial system? Well, one thing you could do is just swap it, as we were told earlier. But I think, and I think like that motivation is the same as the one that I'm talking about, right? Because when you swap it, you're saying, does the rest of the system treat this in the same way, right? That's the right perspective from which to assess similarity. But I think you don't necessarily need to look at the whole downstream system, right? You might just look at what is the next layer doing? What is the next layer sensitive to? What is the next area that receives projections from the activity that you're measuring? What is that sensitive to? So if all comparisons of similarity are from a perspective, then if you're looking at V4 activity or layer 4 activity, then it's the perspective of layer 5, or whatever's downstream from layer 4 that matters. And in the case of the neural network model, it's quite nice because this, quote, perspective, this point of view, whatever it is the downstream system is doing, that is itself a kind of transform that we can apply to two candidates to see if they match, right? Whatever your best model is of the next part of the brain, it's some kind of, linear transformation combined with some kind of brain-like nonlinearity that the actual neurons might apply. That kind of transform is the kind of thing that ought to pick out just those parts of the activation that are actually functionally relevant, while ignoring the parts that are not functionally relevant. So I think that, so when you say that everything is similar in some respects and dissimilar in other respects, looking at what the rest of the brain thinks helps you pick out in an objective, non-subjective way, which are the relevant respects and which are the irrelevant respects, right? It's not just going to be some kind of expression of your prejudices about whether initializations really matter or not. It's going to be whether the brain treats them as different, whether downstream parts of the brain treat them as different or not. So that's the subset that's going to be important for the purposes of the comparison. So here are some consequences of using a kind of a brain-like perspective. It probably isn't going to be just linear because we know that the downstream parts do more than linear things. It's probably not going to be symmetric or ametric, and that's consistent with this old psychological work about similarity judgments. And it's also a pluralist perspective because the right mapping to use is going to depend on which areas we're interested in and what lies downstream. So you might use a different similarity comparison depending on whether you're studying visual cortex or hippocampus. And you might use a different perspective depending on whether you're trying to compare brains or machines. So in the case of the substitution experiments, I think the substitution experiments that were actually done tell us whether, from the perspective of an artificial system, the brain activity is the same as the artificial system's activity. But maybe what we're really interested in is whether, from the brain's perspective, the artificial system's activity is the same as the brain's activity. And that means you need to feed the artificial system's activity into the brain rather than the other way around. So I'm just going to wrap up here, but there's some empirical work from Dan Yemen's lab where they look for a transform not based on these motivations, but just empirically try to find something that does a good job of separating models in ways that they think are intuitively different, in ways that they think are different, while showing models that they think should be the same or the same. They do find something that looks kind of brain-like. So you can ask Aran about that work if you're interested. Thanks.
Thank you, Rosa. So now we'll start the next session where our team members are going to argue that we probably need more data. Our first speaker is Ratan Murty, who's a research scientist at MIT.
Okay, great. Thank you all for being here. So in my part of my talk, what I want to really discuss is a central question, which is what are the kinds of experiments that we really should be running? And my central thesis for what I'm going to talk about is that we need a change of mindset. This is, of course, not easy because our mindset is really, of experimentalists is really driven by many external factors, such as the kinds of reward structures that we have, how much time it takes to run experiments, and so on. But the thing that I want to argue for today is that we, as experimenters, give relatively less importance to the current models or hypotheses that we have out there. And there's actually a historical reason to this fact. For example, we had, in the pre-deep learning kind of era, I would say that we were collecting a lot of phenomena. And this has been really well documented across the history of science. This is a great paper, like you can't play 20 questions with nature and win. If you have not read this paper, I'd really recommend it. But what I really mean by a collection of phenomena is that you have a bunch of neural phenomena. You can think of them as stuff like pinwheels, blobs, selectivity, repetition suppression, things like mirror confusion, and so on. And we were collecting a lot of behavioral phenomena on the other hand, things like holistic processing, backward masking, visual search effects, initial blindness, and so on. Now, to be fair, we did not have good computational models that could even explain these phenomena that we were collecting. And then, as you all know, everything changed. We had a revolution of sorts. We found architectures that were trained on these large corpus of visual data. And then we also found correspondences between early brain regions and the hierarchy and so on. This has been well documented throughout the entire series of talks that we have, so on. And then, as I mentioned before, the progress seems to have slowed down, right? And let me take a while to reflect on the success. So what do these results really tell us about the kinds of data that we were collecting? And for the purposes of this talk, I'm going to be wearing a hat. We are at Oxford, after all. I'm going to wear an experimenter hat. And I want to argue two things, really. The first thing is that previous studies actually give us very important clues about the kinds of experiments that we should be running. And the second clue that I want to actually drive home is that we need different kinds of data now than what we have currently in order to really truly falsify the current class of models that we have. So the central thing that I want to discuss today is that we are really limited by data that we have. So let's look at the first part. I think previous results provide important clues. So the central thing that I want to drive home here is that how good our models are, at least on the metrics that we are using today, suggests something about the granularity of the data that we have been collecting so far. So imagine that you have a two-dimensional axis like this. On the x-axis, you have what I'm going to call data granularity. You can think of this as the difficulty of the data that you're trying to explain, the number of conditions you are testing, and so on. And on the y-axis, you have something called model goodness, which you can think of as, say, explained variance for now. Now, this can have many different functions, but I hope all of you agree that the more difficult data that you collect, the model goodness is going to go down, right? And I think we are here, right? So there's success. We have good predictive models that can explain the kinds of data that we have collected for a very long time, right? But now the question is, do all these models really and truly explain prior experimental phenomena, right? How do we know? Isn't that the goal, right? The goal is that we have our current models, and they have to be evaluated, at least explain all the phenomena that we have been collecting over the years, right? So now here's the thing that I started actually on the side, which was, let's see how many of the large number of cognitive neuroscience studies you can directly test based on current brain data. But the goal is to do this completely zero shot. So you're not allowed to retrain the model on the new data sets from the previously collected data. So our lab is really interested in understanding the neural mechanisms of face recognition, for example. So we took all of these studies. Okay, so the key thing to note here is that each of them is actually a theoretical idea. It's embodying a particular question, right? So let me give you an example. So I'm going to show you one particular study. So this is by Danny Dilks's lab, and they wanted to test the importance of internal face features versus external face features. So you have internal face features, I mean something like this, just cutouts of eyes and nose, and then you can remove all of those internal features, and you have the external features there. And here are the 4 bars that you get from a given particular experiment, right? You get a high response to both the internal features and the external features, and you get a relatively low response to the other two conditions. If you run the same experiment on a computational model of the FFA, you get something like this. So on a qualitative level, you are predicting the kind of pattern that you saw from brain data, right? Now, here's a problem. And this problem has been demonstrated several times by now. We have stuff from Talia Conker's lab, and on the BrainScore platform, almost all models can recapitulate this qualitative pattern, right? So, but can we use these old data to also quantify what the differences are between models? We've been starting to do that now. So on the y-axis now is a mean square error plot. You can extract data from the original studies and see how well models performed. On the x-axis are a bunch of candidate computational models. And what we find is that you can use these previous data to actually start quantifying differences between models. And the more data that you add, you get a large model separation compared to just evaluating models on one single data set, as has been the norm in the field so far. Okay, so you can take all of these phenomena, you can explain all of them qualitatively, but as you start being more quantitative about your predictions, everything starts to fail. So going back, there's success. We can now successfully explain these phenomena. That's great, but there's also a reality check, which is that all of these phenomena are too easy for current models. So we have to now start going beyond these previous data sets that we have collected. And I think that's what will, I think of that as encouragement that will force the models to, we can expand on the kinds of data that we have been collecting for future studies. And so in the remainder of my talk, I'm going to just do this very extremely briefly. I also want to focus on one other thing, which is that we have been collecting too many data sets on visual responses, and that we need to collect data sets that also capture different aspects of the current models. I'm going to give you just one example, which is a case study for now. We already know that ANN models can predict data to a very large degree. We've seen that over the course of this GAC as well. Now, the question is, how do these models actually learn the representations? And now there's some work. I'm going to show you just two of them. Here's a work from Martin Schrimpf and Chengshu Zhang, and Dan Yeamans, and the other by Talia and George, that show that you can have these models trained not on classification, and these models can predict data really well. But notice that there's a kind of disconnect here, right? These are claims about model learning, but they have been evaluated on model inference. And that is for a reason, right? Where is the data? Right? We need to move beyond just collecting data on just visual responses, and think about even collecting data from longitudinal data over how primate neural responses develop across time, behavioral data across human infants, and start to really collect data where we compare neural learning against these data. And with that, I would like to summarize. These are the two main take-away points that I wanted to focus on in today's talk. And I hope you agree with me that what's really limiting us in order to make progress that can completely falsify all the models that we have on the table are actually data. Thank you.
Next up, in this neural data session, we have Jenelle Feather, who's a research fellow at Flatiron Institute.
Hi, everyone. So today, what I'm going to be talking about is how we can use behavioral differences of models to motivate new neural data sets. And I think that this really builds on a lot of what we have seen so far. Specifically, even though many people have talked about the fact that these models are kind of indistinguishable in these neural benchmarks, and it's the case that actually we do have behavioral benchmarks that show very clear differences between our models. And so in that sense, we actually do have ways to tease apart how the model representations are different, even though they are predicting the same neural activity. So some examples of this, the most sort of maybe classic case that people can think of are adversarial examples. So just a quick reminder, adversarial examples are stimuli where small perturbations change a model's classification decision, but these perturbations are imperceptible to human observers. And so this is a place where these models seem to diverge with human perception. However, we actually now have some models that are improved on this type of evaluation metric. So specifically, if you use a type of training called adversarial training, you can find new models that maintain accuracy for larger adversarial perturbations. And so there's very clear differences between the blue line and the black line here. Now, another way that we have noticed behavioral benchmarks which show a clear difference is using the idea of model metamers. And so this was really the focus of my PhD work. So model metamers are two stimuli that produce the same activations at a particular stage of a computational model. So to flesh this out a little bit, we have two stimuli. One of them is maybe a natural stimulus, and one of them, in this case, is a synthetic stimulus, which we're going to call our model metamer. But whenever both of these stimuli, or either of them, are presented into some neural network, in this case, it's a neural network that's trained to do speech classification, we see that at some particular stage, the activations are the same for the input stimulus and for the model metamer. And so what we do in order to sort of evaluate how well models perform on this is we'll generate metamers from each model stage, and then we'll present those metamers to human observers and have human observers do the same classification task that the model was performing on this. Now, if we generate these stimuli from many different stages, in this case of a visual neural network, what we see is that at the very final stages of these networks, we start to generate stimuli that look very different than the original. But note, these stimuli are essentially matched activation for activation at a particular stage of our deep neural network to be the same as this natural image. And so the network is kind of equally confident, you could say, that this image is a bear. Now, if we go and present these images to human observers, we see, sort of as expected from these examples, that humans are not able to recognize these stimuli as the model class that they originally belonged to. And so this, in this case, is evaluated on a 16-way forced choice behavioral paradigm. Now, once again, we actually do have some models where we see an improvement on this type of behavioral benchmark. This example that I'm showing here is actually the same type of model that I was showing before. This is an adversarially trained model. And what we see in this case is that model metamers from these final model stages start to look much more natural to human observers. And additionally, these metamers are more recognizable to human observers if we actually quantify this with behavior. Now, another sort of final case of a behavioral benchmark that people have proposed are just these other types of what people would call out-of-distribution stimuli. And so a lot of what I'm talking about now also relates to what Tal was talking about in the first section. So these out-of-distribution stimuli are, in this case, some of the examples are this natural image, which is a dog, and then there's different types of augmentations that are added to this image of a dog, such that to humans, these are all still classified as a dog. They might look a little bit different, but behaviorally, they're classified the same. And in 2021, Robert Garris and all showed that actually there's quite a wide range of models' performance on these types of out-of-distribution examples. And specifically, they also found that performance on this out-of-distribution stimuli improves whenever larger data set sizes are used. And so using all of these three benchmarks, we can see that actually we can tease apart variations within our models. But what happens whenever we present these or what happens whenever we try and build some of these regression mappings between the model activations and neural data? And so these changes, for instance, adversarial training or training data on large stimulus sets, do not lead to improved neural predictions in IT. And so in this case, seeing some improvement on these behavioral benchmarks doesn't lead to explicit improvement. And so just to sort of hammer this home, I'm going to present some scatter plots here, where I'm going to show the IT predictivity from brain score on the x-axis. And note that I am going from 0 to 1 because this metric is supposed to be sort of corrected for noise. And so I think that we should see that if we have some metric that we think is good, we should at least be able to get 1 on that metric if it's noise corrected. And then additionally, I'm going to be showing this behavioral metric on the y-axis. And let's go through these 3 different behavioral metrics. So first off, these are examples from adversarial robustness. And so what we see is that the adversarial accuracy for these adversarially trained models is significantly better than many of the standard models. But the IT predictivity doesn't really change. And so here, the R-squared value between the IT predictivity and the adversarial accuracy is essentially 0. So we're not seeing any difference, even though we know that the representations are different. Now, in this case, what I'm showing is the same correlation, but using this framework of model metamers. And so on the y-axis here, I'm plotting the human recognition that is measured from model metamer recognition at the committed model stage. So this is exactly the same stage that was used in order to build that mapping to the IT data. And once again, we see that there's essentially no correlation here between the IT predictivity and human recognition, even though we know that these representations are in fact very different in terms of how they mediate human behavior. And then finally, we have this out-of-distribution evaluation, where once again, we see the sort of lack of any sort of a correspondence here, even for these models that have larger training data sets. So all of these 3 behavioral benchmarks really show differences between models, but whenever we then use those types of models in order to predict neural data, we see that this doesn't actually make things worse in terms of our evaluation. So that's nice. These types of changes aren't making our neural predictions worse, but they also aren't making things better. So how exactly do we reconcile this? How do we reconcile this difference between behavior and our current neural benchmarks? And so what I'm going to argue is that we can actually use some of these observed behavioral differences as potential motivation when we're collecting new neural data sets. So let's just go through a very quick thought experiment here. Maybe we measure the neural responses for model metamers from our standard and adversarially trained models. Now, if we do this, we can then use these data for new neural predictions. And presumably, the 2 models then would show a difference in predicting IT responses. But if they don't, it would be very surprising, because in that case, we would think that, well, IT is actually doing something quite different than what our current models our current sort of idea expects, because it's not mediating behavior in this way. Okay, so just to quickly conclude, I also do want to say that all of these behavioral benchmarks, you can also think about them as improved metrics. But in addition to these behavioral benchmarks being improved metrics, I'm also working on this work that Suyan talked about earlier, where we are looking at the geometry of these networks in more detail. And in fact, with some of these metrics, we do see that there are differences between these types of models, in this case, standard and adversarially trained networks that have similar neural predictivity, but the way that they achieve this is different. So thanks to my collaborators on this project.
So our final speaker for today is Radek Cichy from Free University.
Okay, thank you very much for organizing this. Last talk, I was told it's the last talk, no one behind me, so I can take all the time and run over time. No worries, I will try not to do that. So thanks a lot to the organizers. I'm happy to take the devil advocate's position for data today. And I was told to make it punchy and provocative. I will try. So, you know, theory, schmeary, tools, schmools, it's always data that rules. I will try to make it as concrete as possible, but also show up open questions where I think that there are some. So I will be talking about 3 questions. One is, why is actually having more data a good thing? This seems to be self-evident, but you know, we still don't have it, and just knowing that something is good for you doesn't mean that you're going to do it. You know, you should all not eat junk food and go to the gym, and you don't. So why? And it's the same with data. And the second, then, I will try to say, okay, then if we think yes, we need more data, so what kind of data do we need? And then, of course, how do we get this data, and how do we keep it coming? The second point will be a bit more concrete, and like a thought experiment where I try to take you along, and the third one is going to be very open. So let's try to go through a couple of arguments that we should actually all know, but forget why we should have more data and neural data for actually every purpose. And one is just a typical noise and complexity argument. So neural imaging data is always noisy and the dynamics always complex. And you can do whatever you want with all your tools and all your metrics. You can't do away with that. That's like one brute force limitation. And you need more data to get over that. There is no shortcut, unfortunately. So more data can cancel out the noise and capture these dynamics. Less trivially and more tailored to the questions here is the size of the cognitive space argument. The spaces we talk about are huge. We're talking about all visual percepts you can have, or maybe all linguistic expressions for large language models. And if you want to densely capture that space, you can't do it with a couple of stimuli. That's just not possible. You need a huge amount of stimuli, and for that you need a lot of data, because more data will sample the space better. The third one is an argument from history, which says that progress until now was correlated strongly with data availability, and history is future. Meaning, once you have the data, you can do things, and if you don't have the data, you're always stuck and you're always doing the old things. So if I just, as a case study, look at what we've done in the lab, in the days of yore, when we started this, we had suddenly, did a great effort to get 100 images. And that allowed us to compare DNNs with brains, which was great. But now, a couple of years later, we thought, okay, we can't do many things, so we need more images. So for example, Ale Gifford has collected 16,000 images in EEG, which then allowed us to do new things methodologically, like end-to-end DNN training on EEG data. Or Ben Lahner has collected fMRI data to over 1,000 videos, which then allowed us to look at modeling events rather than images, bringing us theoretically more towards what you actually want to explain in humans and how they actually really see the world. And the fourth is actually an aesthetic argument. It's always nicer to manage riches than poverty. So think about all the times when you just couldn't continue, and you had, to look at your p-values which didn't go right, and you just couldn't go deeper and find more convincing evidence for something because the data just wasn't there. So if you just get the data in the beginning, you will have a very nice time afterwards. So I hope now you're all excited and you want to go out and you want to collect data. So how could that look like? So what kind of data do we need? In every question, every field, you will have to make a huge amount of decisions about this. And I will just try to go through a list of decisions for which I have, let's say, made mine. And you can just completely disagree with that, but that's like thinking it through what you would need. So the first thing is species. What species do you want? The Bushido gives the samurai 8 breaths to make a decision. I give you not even half, you don't have to, 8 minutes. So I would say for me, it's going to be humans, because that's what we care about, and the majority of the field works there. And if you want to get it fast, that's the fastest species to get it in. Should it be non-invasive or invasive? No, I think that's easy. It should be non-invasive because it's completely easy and fast to get. You can always get many images in just a college student, but get, let's say, 1 million images in ECOG patient, difficult. Should it be targeted to specific aims, as some people have argued, or should it be good for all aims? And here my position is very clearly it should be good for all, because today's models will be outlived tomorrow. So once we have the data today, it won't be relevant anymore to test the new models. Should it be integrated, so structured like a new thing that we do, or should it be aggregated across different sources where we just take all the data, a huge amount of data that we have, and put it together? And I would say we should integrate it, because then it's going to make the comparison easy and strong. And in the end, should it be good for explanation? Should it be more bound to a theoretical question or good for prediction modeling? And I think it should be bridging both. And how this could possibly maybe be done, I would like to show you. So one idea would be to say, okay, let's say the workhorse that we right now have is naturalistic still images. Let's go more towards the theory where we have collected, as Ratan said, a huge amount of phenomena in psychology, for example, and use very tightly controlled artificial still images as well, where we know the ground truth and we know exactly what we want to do. But then let's also go more naturalistic and ecologically valid down to something like movies, which are closer to what we actually have. And we want these data to be huge because we want to constrain a lot of parameters and models, and we want it to be very diverse. We also want, if you want this non-invasively in humans, it must be multimodal because you can either only get space or time, and we want both, so you will have to have 2 sessions. So that will buy you actually something that bridges theory and modeling and enables all the unprecedented analysis insights and modeling that you also have seen here. Just think about all the analyses that have been made possible in this room and talks that by, for example, people like NSD providing the data. Now, if you calculate it through, is this like a madman's business, or can you actually do it? So if you calculate it through, for about 10 participants, which is 16 sessions each, which is just one semester. In EEG, you can get 1 million images and 30,000 videos, and in fMRI a lot less. But you can get to 1 million stimuli, which is just so much more than we currently have, and kind of going into the ballpark of ImageNet. So the third thing, how do we get this data and keep it coming? I think, if you looked at the last slide, how much effort it would actually take to get a huge data set of 1 million images, a single lab can do this. It's difficult and it takes some time, but if you just go out and you spend 3 months on this, you can have this data, so it's possible. So a single lab can always make a significant contribution, but many labs together can make, of course, much larger and stronger contributions, and there's a lot of models already out there that try to bring people together in different ways. So there is BrainScore, there is challenges, like Ergonauts, there is generative adversarial correlations, like Cogitate, there is the Things initiative, which is an own thing in itself, and there's something that maybe in our field started this more like an international brain lab. And they are all different measures that you can use. The only thing I would like to say is that when you choose a collaborative structure, and I think we have to do this to get really large data, this also decides who makes the decisions about what data is recorded and how transparent and open it is, and how it's distributed. So I think when we talk about data acquisition, it's as important as what data we get, as how we get it, and how sustainable and open we make it to others. And with that, I would like to end and thank the lab.
Okay, so unfortunately, we don't have time for Q&As. So thank you all for attending the workshop and for actively participating on the Slido platform. Even though we were time constrained, we'll really try to engage with those questions offline. And if you have any questions or ideas that you want to share with us, just feel free to reach out during the rest of the conference. Thank you all.